Hello everyone. Today we'll start with the topic apoptosis. Apoptosis is the pathway of cell death which is induced by a tightly regulated intracellular program in which cells are destined to die. Basically, apoptosis is the cell suicide. What happens is that they are a caspase. So, what happens is there is a regulated intracellular program which activate the enzyme, namely caspase, right? So they degrade their own nuclear DNA and cytoplasmic protein. So basically, now these they are called as the apoptotic bodies, right? So now here the cell is phagocytosed. There is no leakage, as there in necrosis there was leakage. Here is no leakage, so there is no inflammation. So it is the way of elimination of unwanted cells and those cells which are damaged beyond repair capacity of the cell. So apoptosis generally includes single cell in contrast to necrosis that usually involve a group of cells. So it is the programmed cell death. It is genetically programmed. So it is programmed cell death. It is genetically programmed and it is an energy dependent process. So if we talk about the mechanism of apoptosis, which is also important. So basically the apoptosis whole topic is very important. So we'll divide the mechanism into two parts, that is the extrinsic pathway and the intrinsic pathway. So here the extrinsic pathway is subdivided into two pathways, that is initiation and the execution. Likewise, intrinsic also have the two pathway, initiation and the execution. So firstly, talking about the extrinsic pathway for the initiation phase, let's see that first. So talking about the extrinsic pathway first for the initiation phase, let's see first the initiation phase. So here it is initiated through specific receptors. So those specific receptors, they are called as the death receptors. So we have two. First one is the Fas protein called as CD95 and the other one is tumor necrotic vector receptors, right? So what happens basically in this is that here we have the death receptors, right? So these are the death receptor, we can say it as Fas or CD95 receptor, and here there is a Fas ligand, right? So what happens here is that when this Fas receptor, death receptor, right, it binds with this Fas protein, this receptor, right, Fas protein binds with the death receptor. So these multiple receptors, they come together, right? They will come together and the cytoplasmic death domain is formed. They bind with this, they come together, cytoplasmic death domain is formed. So this will activate, what will this do? This will activate the procaspase 8 and 10 to active caspase 8 and 10. So this was the extrinsic pathway initiation phase. So now talking about the intrinsic pathway initiation phase. As in the extrinsic pathway initiation phase, we did those receptors, and in this intrinsic pathway initiation phase, what is there? The stimuli. So here the stimuli is the DNA damage. So what happens here is that if, let's suppose there is a DNA damage, right? So this signal, right? So what happens is this, if this DNA damage is beyond the repair, so here the signal will go right here, and the anti-apoptotic molecules, right? So anti-apoptotic molecules, for example, BCLX, MCL1, BCL2, right? So this will be replaced with the pro-apoptotic molecules, right? So they will get replaced by this. So what are the pro-apoptotic molecules? These are the BAX, BIM, PAD, PAC, right? So they'll get replaced by pro-apoptotic, and this will increase the mitochondrial permeability. So we know that here is the presence of the cytochrome C. So there will be release of cytochrome C in the cytoplasm. So what this will do is this will activate the APAF1. Along with this, there will be activation of procaspase 9 into the activated caspase 9. So we have the activation of APAF1 along with procaspase 9 into caspase 9. So what happens is these are replaced by the apoptotic. So these anti-apoptotic molecules, they prevents apoptosis, and here we are studying the apoptosis, how it is occurring. So if these prevent, they are converting into this, means these they causes apoptosis, right? Right, and they have shorter life and they have longer life. Okay, for example, I told you BCL2, MCL1, and here we have done BAX, BAD, right? So talking about the execution phase for the extrinsic and intrinsic, both are the common. Okay, common pathways are there for the execution phase, as the initiation phase has different ones, so we discussed before. Now we'll see the execution phase, which is common for both the extrinsic and intrinsic, right? So what is there in this? So this execution phase, this is the convergence point, right? This is the convergence point for both the extrinsic pathway and the intrinsic pathway. So as we discussed, in the extrinsic pathway, there was activation of caspase 8 and 10, and in intrinsic there was the activation of caspase 9, right? So now in this execution pathway, these both will lead to the activation of caspase 3, 6 and 7, right? So sequentially this will activate all the other caspase, and the caspase deplete the cytoskeletal and nucleus matrix proteins. So there will be cell degeneration and the apoptotic bodies are formed. Now talking about the morphological changes in apoptosis. So what is there? This is a cell with the nucleus, right? So firstly there will be the uh, sorry there will be the shrinkage of the cell, right? First is the cell shrinkage. Second, that this will be pinkish, right? Eosinophilia. Then pycnosis is there. So what is pycnosis? Pycnosis is the condensation of the nucleus, right? After the pycnosis there is karyorrhexis. Now what is karyorrhexis? This is the fragmentation of the nucleus. So pycnosis was the condensation of nucleus, karyorrhexis is the fragmentation of nucleus, and then there will be the fifth, that is yeah, fifth, there will be formation of blebs, right? And then lastly there will be the formation of apoptotic bodies. So this was all about the apoptosis. Thank you.